This beverage on the right is something that I call cran-orapple juice. It's kind of a mixture between, you can guess from the name, cranberry juice, orange juice, apple juice. I kind of made this beverage so I know what made it up, but if we didn't know what made it up, one possibility that we could use to figure out the different ingredients that make up this mixture would be, you guessed it, chromatography. Chromatography, it's been around for a little over a century, first being used by Mikhail Tsvet, this gentleman here. He's a 1906 Russian botanist who used it to separate plant pigments into bands of color. The name chromatography literally means color writing. We'll see how that fits in later, but odds are, if you've taken general chemistry, you've done the lab, and it looks a lot like this right here. The general idea of chromatography is that components in a complex mixture will be separated across a stationary phase by a mobile phase. There are other types of chromatography than paper chromatography, like you can use gas chromatography, but with paper, the mobile phase is the liquid that absorbs upward from the bottom, where various mixtures are placed beforehand, ready to be carried up and strung out and kind of separated there. By process of elimination, the stationary phase then must be the chromatography paper. Depending on the size and polarity of the various components in the mixtures, they will find their stopping points at different distances from the start at different speeds. So components move rapidly if they're polar and small, and the slower ones are typically not as polar or not polar and are larger in size. Once they've met their stopping points, the mobile phase sort of drops them off at their stop and continues on as far as the stationary phase goes. If a mixture is too polar, it will move up too rapidly for much effective separation. It's kind of a mess there, and that leads to inconclusive results. So the use of paper chromatography is to separate complex mixtures into components. If we are looking for specific components, we will have a control solvent with known components and compare those with the mixture being tested. If components in the tested mixture line up with components in the known solvent as far as the distance traveled up on the paper or the stationary phase, we can then more accurately say what makes up the tested mixture. So we would see bits of cranberry and orange and apple when we test our cran-orapple juice. So that's the basics of chromatography, and we have old Mikhail to thank for that.